Chalo. First, gene, maybe definition written. So, what is gene? Structural and functional unit of the inheritance. Remember that. Structural and functional unit of inheritance is called as a gene. From one generation to another generation, gene actually transfer. Now, gene control the character. Remember that. Gene control character. But one gene is formed by pair of allele. Pair of allele. So pair of allele will be together to form one gene. Gene is responsible one character. Now character, it may be phenotypic character, phenotype. It may be genotype. Remember that. Phenotype or genotype. These are the kind of character. Now phenotype character means which is outside visible. I am tall or dwarf? Tall. So visibly I am tall. But you don't know I am pure tall or mixture tall. That you can't say. Getting my point? You can't say on that particular part. So whatever is visible, that visible character is called as a phenotypic character. Genotype means inner genetical character. Okay. Now, just starting from the beginning, we know about the Mendel. Mendel birth year was 1822. 1822 was Mendel birth year. And understand that during that time, actually the study time, NEET question came 1856 to 1863. That is a study time when he studied on the pea plant. He studied on the pea plant. Remember that Mendel was not a biologist. Mendel was a mathematician, especially statistician. Now, if any mathematics teacher comes to the biology class and discuss about the biology, obviously biologists will laugh. So same happened with the Mendel. Mendel work was not recognized. So why his work was not recognized? Because his background, it's not from the biology background. Statistician. So no one recognized his work. And whenever any scientific work actually happen, any research type of job, that need to be published in international journal. So international recognition comes. He published in local journal. Okay. Clear? So local people know about that and they have no such headache about this work. So outside people don't know about his work. So during his lifetime, his death year was 1884. Death. This is birth, this is death. Okay, these two not given, but given this one and question came from this part when he studied on the pea plant. Clear? So once it done, in 1900, from the different parts of the world, 3 different scientists, they rediscover his work. That okay, wherever we are working, that work is already discovered early by Mendel. So Mendel work rediscovered by the 3 different scientists. Okay, he was a priest in a church, and in the backyard of the church there are lots of gardens. So in that garden there was a lots of pea plant. So what he have observed, major pea plants are tall, some are dwarf. Major pea plant having the flower color violet or purple. Some are white. So this major character, whatever he have observed, he discussed them as a dominant character. In a population, that character is maximum, that character is dominant. What is recessive? Less number of character present in a population. Majority of us is a tall. Maybe some of you are dwarf. So in a population, that actually happens. Is it clear or not? Now, as I told you, that pair of allele. So how we have to denote the allele? As I told you, the character. So let's say the character of the plant is a height. Height. So dominant character, how it will be dominant? Because as I told you, in population, in a particular population, the major character of this. That's why it's a dominant. So dominant is a tall. In a population, least number of character is a dwarf. So that is recessive character. That is dwarf. Clear? Mendel know about this fact. But Mendel don't know how this is happened. Mendel have no such idea regarding gene. Understand that 1856, during that time, microscopic idea was very, very elementary level, basic level. No such idea was there. Clear? So whatever Mendel told us factor. Remember that. Whatever Mendel discussed about the gene as a factor, NEET question. This is factor. Later it was discovered whatever Mendel discussed as a factor, that is actually gene. So Mendel discussed gene as a factor, that some factor is responsible for the character. Later, other scientists, Johansen, there is one scientist who termed gene. Mendel did not term gene because Mendel has no such idea. This is clear? How dominant and how recessive concept came. Now, understand that how we will put the allele. Major, maybe, that's why I am recording. Valid proof will be there. 955. Till now I am saying, few of you will 1000% do the mistake. Listen me very carefully. How to denote the allele? Always the dominant character. First letter we will take. Tall, capital T, dwarf, it's not D. It's a small letter of the dominant one. Getting my point or not? Always how to denote the allele? Dominant character, first letter will be in the capital for the dominant allele. The same letter will be used in the small one for the recessive. So one is capital T, another is small t. These are the two characters we have. Capital T for dominant, small t is for recessive. So I am the representative, capital T. Let's say you are the representative, small t. Imperial have capital T and small t. How many combination is possible? 3 combination. Capital T, capital T, capital T, small t, small t, small t. Clear? Now, if that is the scenario, so what is the character will come? Tall. This is also tall. This is dwarf. Because 2 tall character there. This capital T is also tall. This capital T is also tall. Clear? So they will represent tall. That's why the phenotype character is a tall. You will see visibly that is tall. But whenever we go to the genotype, that genotype character is a pure tall or homozygous tall. Pure or homozygous, same, or homozygous tall. But here, this one, remember that whenever the mixture tall will be there, Mendelian first law is applicable here. That is a law of dominance. So here we get the first law of Mendel, law of dominance. So what is the law of dominance? Always the dominating allele will dominate over the recessive allele. Always the teacher, capital T, will dominate over the student, small t. Outcoming character will be the character of the teacher. Always remember that. So this is mixture tall, heterozygous tall, heterozygous tall, mixture tall. This is homozygous dwarf, recessive character. Whenever it will express, it will easy to express because both are recessive. Small, recently viral in Instagram. So if C comes in the class, we can easily say, what about her height? Small t, small t. We have no doubts. No doubt we can easily say small t, small t. Getting my point or not? Have you seen her mother? If you've seen her mother is also small t, small t. Getting my point or not? So one of the small t will come from the mother side. So sure sort, she have get one of the small t from her mother. But till now I have not seen her father, so I don't know. Maybe her father, if even tall, understand that, but that is heterozygous. So thereby easily we can detect the father, that is pedigree. That is called as a pedigree. Whatever I am talking about right now, that is called as a family history analysis. We know about mother visibly dwarf, her character visibly dwarf. So obviously one of the small t will come from mother side, one of the small t need to become from father side. So father never be homozygous. Homozygous tall never possible. That's how we can predict, and that is a way to solve the pedigree analysis. So this is homozygous dwarf or recessive character. Understand, na? So this one is a NEET question, factor is a NEET question, remember that. These two are the NEET question. Year of study, in which year Mendel have studied, and the factor. Mendel discussed the factor. Always the confusion character, which one is bit confused? Dominant one. Recessive character, there is no confusion. And this entire concept will be used for two chapters. One, evolution. There we have to solve some mathematical problem. Heidelberg equilibrium. Another utility here, another utility in pedigree. Monohybrid cross. Simple cross, same T version, just parental type. Here, every left side write up is required. Every. I'm not talking about to solve the board question. Here we have to solve MCQ. And remember that when the monohybrid cross, dihybrid cross will come in a written version, it will be very much tough to detect. Very much. So P for parent, if you don't know, write down the full form. Here, capital T cross with the small t, small t. So parent will form the gamete. Now, for the gamete formation, second law of Mendel is applicable. Law of segregation. Law of segregation. So how the law of segregation will be applicable? Here capital T will be separated, comma, small t, capital T will be separated, cross. This small t will be separated, comma, this small t will be separated. Always remember this process. Forget about NCERT, F1. Why it will be required? Tricks is applicable. At the end of the day, we have to apply the tricks. Tricks only will be applicable if you know the basic process. This is the step-by-step process. So parent will form the gamete. After the gamete, F1 generation will be produced. So how many gamete here? Tell me how many gamete form? 2 gamete. How many gamete will be here? 2 gamete. So 2 by 2 we have to take 3 by 3 checkerboard. Remember that question also came in the NEET. That Punnett square or checkerboard. Okay. So 2 capital T, small t, small t. So obviously this box how we will fill up? One of the T will come here, another T will come here. So I have option that capital T, small t. So who will come first? Always the capital one, then the small. Capital, small, capital, small, capital, small. So what is the phenotype ratio? PR for phenotype ratio. So visibly they are all are tall. visibly they all are tall. So tall is 4 is to 0 is dwarf. There is no other character. Okay. Now is F1 cross. F1 cross. Remember that F1 cross is called as a selfing. Selfing. So what is the meaning of selfing? Self or selfing meaning that they are the child, belong from same parents. Clear? So they are brothers and sister. Clear? So if we have to proceed to the next generation, we have to give them the marriage. That's why the selfing term. Selfing means within family marriage. F1 cross is termed as a self or selfing. Okay? Now, same, you will take again F1 cross. Now F1 gamete will be produced. This capital T will be separated, comma, this small t will be separated. Cross with this capital T will be separated, comma, this small t will be separated. So again 2 by 2 gamete. So we will take 3 by 3 checkerboard, F2 checkerboard. So capital T, small t, capital T, small t. So here capital T, capital T. Next capital T, small t, next capital T, small t, and the last one is small t, small t. So what will be the phenotype ratio? Phenotype ratio. This is tall phenotypically. This is also tall phenotypically. This is also tall phenotypically. But this is dwarf. So how many tall here? 3 tall is to 1 dwarf. 3 tall is to 1 dwarf. Understand? Now, genotype ratio. Genotype ratio. So let's write down all capital T is to capital T, small t is to capital T, small t is to small t, small t. So phenotypically, this one capital T, homozygous tall. These two capital T, small t, heterozygous tall, and the last one homozygous dwarf. Homozygous dwarf. What is the ratio? 1 is to 2 is to 1. Remember that. So standard Mendelian cross. Remember that. This is entirely standard Mendelian cross. For standard Mendelian cross, F2 generation phenotype ratio is 3 is to 1. F2 generation genotype ratio is 1 is to 2 is to 1. Clear? Understand? Now write down problem. Round shape of seed. Round shape of seed is dominant. Round shape of seed is dominant over wrinkled shape of seed. Round shape of seed is dominant over wrinkled shape of seed. If heterozygous round shape of seed, if heterozygous round shape of seed is self-cross or is itself, and 1600 1600 seedling 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 produce in that cross how many seedling will have parental phenotype? How many seedling will have parental phenotype? How many seedling will have parental phenotype? Quickly solve this. So what is given? That round shape of seed is dominant over the wrinkled shape of seed and heterozygous round shape of seed. So what is that? Heterozygous round shape of seed. So capital R small r is self-cross. Self-cross means this. So this is what? Simple F1 generation. This is F1 cross. Is it or not? This is simple F1 cross. So how this F1 cross will be possible? Obviously they have parent. So who are the parent? Capital R cross with the smaller. Then the F1 generation will be capital R smaller. So F2 generation will be capital R is to capital R smaller is to capital R smaller is to smaller is equal to how much? 1600. Those who do not understand, just check. Is equal to 1600 because total 1600 seedling produce. So total 1600 is this. So how much? How many baby? 4. So every baby having probability is how much? 400. Because 4 4s are 16. Now they are talking about how many seedling will have the parental phenotype. So how many of them will look like their parent? So who is the parent? F2 parent is F1. F1 character is round. So we have to find out how many round. This 14, 400, 400, 400, 12. This is in details. But in one line, how we can solve? Total number of seedling 1600. That is the total number of seedling. They are talking about how many of them will look like their parent. Parent character is a dominant phenotype. So total number of child in F2 will be 4. 4 maximum. Out of the 4, how many dominant? 3. This is in one line. That you have to understand how you will solve. Clear? Very easy. Now you go that process. If you don't know, if you are able to understand, okay, chalo. Total number of the member here is 4. So out of 4, 3 is dominating character. Because round is a dominating character. So ratio to will be same, understand that. Alphabet will change. Capital letter will be there. In place of capital T, capital R will be there. In place of small t, small r will be there. Ratio will be same, each and every single area will be same. Understand or not? Clear? Done, na? Monohybrid cross related problem done. Please practice. Change the alphabet, change the character. Total 7 characters studied by Mendel. Total 7 contrasting characters studied by Mendel. So, in your house, follow the same process. Do not step jump. Now, check it very carefully. Check it very carefully. Mendelian first law. Mendel first law. Law of dominance. Mendel second law. Law of segregation. Now Mendel third law. Third law is only applicable from dihybrid cross. Remember that, and onwards. But third law is not applicable from monohybrid cross. That is the law of independent assortment. Law of independent assortment. Check how the law of independent assortment is applicable. Let's say the situation is the given situation is capital A, small a, capital B, small b, cross with the same. That is the given situation. How many genes are present here? How many genes? 2 genes is present. Remember that. Here 2 genes because 2 alphabet. Whenever 2 alphabet, that will stands for 2 gene. This is 1 gene, A gene, this is B gene. 2 gene. Now, law of independent assortment, how it will be applicable? Listen my explanation very carefully. This is one allele of number 1 gene. This allele, capital A allele of first gene will be independently assorted with capital B allele of the second gene. So gamete will be capital A, capital B, comma. Clear? Just see how I am solving this. This is law of independent assortment. Now, same allele of the first gene will be assorted with the next allele. That is capital A, small b. Now, sir, can I write small b capital A or capital B capital A? No. As the I cannot write this in place of this. Why? Because the first gene is A gene. That's why I have written A first. Clear? I cannot change the position. Let's say right now here is a biology class. Who is the capital B? I am the capital B. You are small b. This is room number 4. That is room number 5. Let's say this is room number 1, that is room number 2. There is a capital C gene is also there. C allele, capital C allele, small c allele is also there. So who will come first? We will come first. Then they will come. Clear or not? Okay. So how the gamete formation will be there? If 2 gene is present, listen me very very carefully. If 2 gene is present, every member of the gene will be present in the gamete formation. If 2 gene is present, every member of the gene will be there. See, minimum A1A is here, minimum 1B is there. We cannot form the gamete in between of us because we are biology gene. Biology gene cannot form the gamete between the biology. That means within gamete formation is never possible. Then how we can form? capital B will be separated, small b will be separated. By that way you can form. That is a monohybrid situation. But we are doing a dihybrid cross. For dihybrid cross, we have to combine. We have to take one member from here, another member from there. Then we can form the gamete. If 3 gene is present, A, B, C, so every member will be there. 1A, 1B, 1C. If A, B, C, D gene will be there, then same. A, 1A, 1B, 1C, 1D will be there. One gamete will form. Getting my point or not? Okay. Now, same will be applicable. Small a with capital B. Small a with capital B. So, why small a coming? Because first gene. Okay. Next. Small a, small b. Clear? In the same way, we will form the gamete there. Same. Write down this process. This is law of independent assortment. How the allele is being assorted with the another gene.